This is the process of DNA replication. This is the initiation phase. DNA replication starts when a large protein complex, the pre-replication complex, binds to the origin of replication. In E. coli bacteria, there is a single circular chromosome. DNA is unwound by an enzyme called helicase. It unwinds DNA by using energy released from ATP hydrolysis. Two replication forks are formed from the ori and proceeds in both directions. Single-stranded binding proteins bind to the unwound strands to keep them from reassociating into a double helix. Topoisomerase I surrounds the unzipping strand and relaxes the twisting that might cause damage to the unwinding DNA.
This is the elongation phase of DNA replication. The enzyme primase binds to the template strand and synthesizes a primer, which is a shorter starter strand of RNA. When the primer is complete, primase is released and the whole primer is degraded. DNA polymerase binds to the growing strand and synthesizes new DNA. DNA polymerase needs a free 3' OH group, which is made by the RNA primer, 5' to 3' on a growing strand in order to add new nucleotides, which are deoxyribonucleotides, one at a time via phosphodiester bond. This enzyme elongates the strand by adding new nucleotides and recognizes the different shapes of the 4 nucleotide bases and binds the bases through hydrogen bonding. The DNA protein clamp binds to the DNA polymerase DNA complex, keeping the enzyme and the DNA associated tightly with each other so it can polymerize up to 50,000 nucleotides before it detaches. Without the clamp, only 20 to 100 phosphodiester linkages can be made. The leading strand is the newly replicating strand that is oriented so that it can grow continuously at its 3' end as the fork opens.
The lagging strand is oriented so that as the fork opens up, its exposed 3' end gets farther away from the fork and an unreplicated gap forms. The lagging strand moves in the opposite direction of the leading strand, making what is called the Okazaki fragments, which are backward stretches with short gaps in between them. Okazaki fragments require its own RNA primer to be synthesized by the enzyme primase. DNA polymerase III adds nucleotides to the new Okazaki fragments only at the 3' end, continuing until it encounters the primer on the previous Okazaki fragment. DNA polymerase I hydrolyzes the primer and replaces it with DNA, but there is still a gap between the two nucleotides. DNA ligase catalyzes the formation of the phosphodiester linkages that finally joins the two Okazaki fragments together. This completes the synthesis of the lagging strand in bacteria. This starts the process of termination in DNA replication. Topoisomerase II will cause the strands of DNA to separate from each other and DNA is fully replicated.